Today, we're stepping into the lab to tackle a pretty huge challenge. How do you study something you can't even see? We're going to explore the fundamental techniques that microbiologists use every single day to pick out one single life form from a world that is just teeming with trillions of them.
I mean, think about it. Microbes are literally everywhere. In the soil, in the air, on your skin right now, and they're all mixed together in this giant, invisible soup. So how in the world does a scientist grab just one specific type of bacteria from that massive crowd to figure out what makes it tick? Well, that's the big question we're going to answer.
So here's our game plan. First, we're going to get a handle on this invisible world. Then, we'll get into the kitchen and see how to cook for microbes and check out their special toolkit. We'll cover the one rule to rule them all, and finally, we'll put everything together to isolate what's called a pure culture.
Okay, first things first. You have to appreciate just how crowded the microbial world is. They are everywhere. Our food, our water, inside of us, it's a massive, jumbled-up population. So the #1 job for any microbiologist is to figure out how to untangle that mess and study them one by one. And the way they do that is by creating a pure culture. This is the absolute goal. It's a sample that has only one type of microbe in it, with zero contamination from anything else. You know, whether you're trying to figure out if a certain bacteria causes a disease or if it produces a new antibiotic, you have to get it by itself first. So how do you grow something you can't even see? Well, you've got to feed it, right? In microbiology, we call this food a culture medium. It's basically a custom-made, nutrient-packed meal designed just for microbes. So let's take a look at the menu. You've basically got two main options. On one end, you have a liquid broth. It's like a nutrient soup, and it's fantastic for growing a huge number of bacteria really fast. But if you want to separate them, you need a solid medium. This gives each microbe its own little patch of real estate to grow on, which is totally essential for isolation. So what's the secret ingredient that turns that liquid soup into a solid surface? It's this incredible stuff called agar. Now here's the wild part. The microbes can't eat it. Agar is just a complex carb from seaweed that acts like gelatin in Jell-O. It creates a firm, stable platform for them to grow on, but it's not actually part of the meal. And this right here is agar's superpower. It melts at boiling, but it doesn't turn solid again until it cools all the way down to 40 degrees Celsius. Now, why is that so important? Well, it means you can grow microbes that thrive at human body temperature, that's about 37.5 degrees Celsius, and the plate stays perfectly solid. It's not going to melt into a puddle in the incubator. Pretty clever, right? And scientists get creative with how they shape this solid media. You've got your standard agar plates, think Petri dishes, which give you a big, wide-open surface that's perfect for separating out different microbes. Then you have agar slants, which are made in test tubes to save space when you just need to keep a pure culture alive. And finally, you have agar deeps, which are used to see how bacteria grow based on how much oxygen they need, since not much can get to the bottom.
All right, we've got the food all sorted out. Now let's look at the tools of the trade. I mean, to handle these invisible organisms, you need a very specific set of equipment.
It really boils down to three main categories. First, you've got your cultivation chambers, like an incubator. It's basically a warm oven that keeps the microbes cozy at their favorite temperature. Then you have your culture vessels. That's your Petri dishes and test tubes that hold the food. And finally, you have transfer instruments, like wire loops and needles, which are what you use to physically move the microbes around.
And speaking of tools, here's a quick and really important safety note. You might find this hard to believe, but scientists used to use their mouths to suck liquids up into pipettes. Yeah. Today, that is a huge no-no. It is absolutely forbidden. We use mechanical pumps for everything, a simple but critical rule that keeps you from, accidentally drinking a culture of dangerous microbes.
Okay, we have our microbial meals, and we have our specialized tools. But none of that matters, absolutely none of it, without mastering the single most important concept in this entire field, sterility. There's this saying in microbiology labs that sterility is the hallmark of successful work, and it is 100% true. Sterility means making something completely, totally free of all life. If you don't, your tools and your media will just get contaminated by whatever's floating in the air, and you'll never, ever get that pure culture. So how do you achieve this?
through something called aseptic technique. Think of it as a set of careful, deliberate moves, like sterilizing a wire loop in a flame before you use it, or only cracking open the lid of a petri dish just enough to work. It's all about creating this invisible, sterile workspace to protect your experiment and, just as importantly, to protect yourself from your experiment.
Okay, let's put it all together now. We've cooked the food, we've gathered the tools, and we've mastered the technique. Now, finally, we can achieve our goal, isolating a pure culture.
So you start with your perfectly sterile agar plate. Then, using your flawless aseptic technique, you take a sample with a mix of microbes and you spread it out across the surface. Then you pop it in the incubator at just the right temperature, and you wait. After a day or two, you come back and see these separate little dots of growth.
And this is the magic of the whole process. Each one of those little isolated dots, we call it a colony, is a pure culture. It all started from one single invisible cell that landed on that exact spot and then divided and divided until it became a visible mound of millions of identical clones. You've done it. You have successfully picked one species out of the crowd.
But really, this whole painstaking process is just the beginning. Now that you have that one microbe all by itself, the real science can start. Now you can ask the big questions. Does it cause a disease? Can it produce a life-saving medicine? Can it help us grow food? Getting the pure culture isn't the end of the story. It's the key that unlocks a whole new universe of discovery.